So on Tuesday, we finished up talking about essentially the different steps in transcription in both prokaryotes and eukaryotes. And now we're going to talk about the mechanisms by which transcription is regulated. So we'll just talk about a couple of different mechanisms. There are many mechanisms out there because transcription has to be regulated. We can't have, any gene turned on at any one time. It has to be, there must be a distinct subset of genes are turned on to, do what that specific cell needs to do in the body. So one way that there is gene regulation are by what's called specific transcription factors. So when we first learned about eukaryotic transcription, we had talked about how there's these TF2 letter, right? TF2A, TF2Bs, etc., that they will bind to the promoter and form this pre-initiation complex with RNA polymerase to then assemble what's required for starting transcription. Those are what's called the general transcription factors because they are required at each promoter. There's what's called specific transcription factors because they only bind to specific promoter regions to then regulate a specific subset of genes. So one example, so the amount of transcription from a particular promoter is regulated by these proteins that bind specific DNA sequences, and then they're going to recruit proteins to stimulate or block transcription. So an example is GAL4. So GAL4 is a specific transcription factor that binds to DNA. It actually binds into the major groove. And it is a gene that binds to this, what's called a upstream activator sequence. So this represents an enhancer element that is upstream of the promoter. So it binds to GAL4. It then recruits other proteins to the promoter to then stimulate the transcription of these genes that regulate galactose metabolism. And so many transcription factors, they bind specific DNA sequences. And again, most transcription factors, they actually bind in the major groove of the DNA. Why would they bind into the major groove, do you think, versus the minor groove of DNA? It's bigger, yes. So why would that make a difference? It's right, yeah, because there's more space in the major groove, it's easier to actually access the bases and read the bases. So, okay. There's also regulation, of course, by epigenetics. You guys knew I was going to talk about that. So histone modification and DNA methylation is also going to regulate gene transcription. And this regulation either occurs by regulating the structure of DNA, really the structure of the chromatin, and its accessibility of that DNA to the transcriptional machinery, or these modifications, they are bound and recognized by these epigenetic proteins called histone reader proteins, which they have these binding sites that specifically recognize an epigenetic mark, a specific covalent modification on the histone tail to then regulate transcription. So just an example, again, how epigenetics can alter chromatin structure. Remember that chromatin is very, very condensed, and it has a very repressive structure. So the DNA is very inaccessible if it's lacking histone modification. So a gene tends to be turned off in these sort of closed repressive structures, and that certain modifications, for example, histone acetylation, can cause the decompaction of chromatin. So the DNA will then kind of become more accessible, and it's easier for transcription factors to be able to come in and access that DNA. It's easier for that transcriptional machinery to come in and bind to the promoter and then turn the gene on to start and initiate transcription. Now, why do you think that lysine acetylation specifically increases DNA accessibility? Okay, lysine acetylation? Mike?
We talked about how the lysine has a positive charge
Mm-hmm.
on the tail of the histones, and
Mm-hmm.
so acetylating it would remove that positive charge, and therefore DNA would be able to spread out more as the negative charge would be neutralized.
Right.
Is that right?
Yes. So pretty much, yes. So essentially, remember that DNA has a negative charge. The tails of the histones have lots of the basic residues that are positively charged, like lysine and arginine, and that there's an electrostatic interaction between the DNA and the histone tails that allows the chromatin to compact very tightly together. When you acetylate the lysine, it neutralizes that positive charge, and so you no longer have the ionic interaction, and the negative charge of the DNA now is more repulsive, and so the DNA or the chromatin is going to spread out. So yes. Okay, and then we're going to talk about this idea of these histone binding domains. So there are these proteins, we call them reader proteins, but they are proteins that they read and bind a specific modification, and it's very similar to how an enzyme will interact with its substrate. And so once that histone reader protein is bound, it will then recruit a transcription factor to the chromatin, and it'll sort of interpret that modification to either turn a gene on or turn a gene off. And this is just kind of an example of a specific histone reader protein. It's called ING2. This is the structure of ING2. It has what's called, you don't have to know this in great detail, but it has what's called a PhD finger, and it binds to lysine 4. I'm trying to see where it is on here. I'm having a hard time seeing. It's right here. So on the histone tail, it binds specifically to lysine 4 that's trimethylated, but you can see that the active site of the protein is very similar to an enzyme where it's this cleft that interacts with the histone through non-covalent interactions. So just kind of an example of how, again, a histone modification or DNA methylation can regulate gene transcription through sort of these histone reader interaction. There are these proteins out there called it's called methylcytosine binding protein, so it binds methylcytosine. We now know, right, that methylation of DNA is generally associated with gene silencing, with gene repression. The reason why that happens is because when a promoter of a gene, so the cytosines, become methylated, that acts as kind of like a chemical signal where it's going to recruit this protein called MECP2. It's a methyl binding protein. It specifically binds to the methylated cytosine. It then recruits other enzymes to the promoter to then turn a gene off. Okay, so it's kind of acting as sort of these readers tend to act as sort of recruiting complexes. They're going to recruit in other proteins to then cause a gene to be silenced or activated. Okay, another mechanism of regulation are small interfering RNAs. How many of you have heard of small siRNAs in your classes? Maybe genetics or cell molecular? Okay, so siRNAs and microRNAs. These are one of the classes of the RNAs that are transcribed, and they have regulatory functions in terms of regulating other RNAs. So sRNAs, they are transcribed by certain regions of the genome, and they tend to form a specific hairpin structure, so it'll complementary base pair with each other, forming a double strand of RNA. There is a protein called dicer, which will then bind to the double-stranded siRNA or microRNA molecule and essentially chop it into a smaller portion. So these are usually about 15 to 100 bases, depending on if it's an siRNA or microRNA. And that siRNA or microRNA, one of the strands of it can actually bind to an mRNA, so it'll complementary base pair to an mRNA, and this complex called the RISC complex will then bind to the sRNA that's hybridized to mRNA and essentially cut the mRNA into little pieces. So it essentially destroys the mRNA. So it acts in a way as sort of a way to cause gene silencing. So that way there's less mRNA transcript being made, therefore less protein being made in the cell. So that was this figure that was on the other side of the handout. Okay, so now we're going to talk about RNA modification after transcription of the primary transcript. Remember, the primary transcript is sort of that initial RNA molecule that is transcribed by the transcriptional machinery, and it is not the final mRNA transcript that will then be translated in the cytoplasm of the cell. So it has to undergo different modification. So the primary transcript is the RNA molecule prior to any modification. Remember that there's there are at least 3 types of mRNAs that are modified. So there's there's at least 3 types of RNAs that are modified. So there's mRNA, tRNAs, and rRNAs. And they're modified by specific enzymes, and how they become modified it's different between prokaryotes and eukaryotes. And so there's essentially 3 types of modification. There is trimming of the transcript. So usually there are leader and trailer sequences that are cut off, and there's also the removal of introns. There is additional terminal sequences as well as base modification. So there can be, for example, methylation of the base. And once the transcript has been completely modified, then this is going to produce the mature transcript. Okay, so let's talk about tRNAs. Modification of tRNAs occurs in both prokaryotes and eukaryotes. So tRNAs, they're transcribed into kind of one long sequence, and then they're going to sort of fold up into this stem loop structure, and then they're going to be trimmed in the nucleus, where they're going to be trimmed at the 5' end as well as the 3' end. At the 3' end, there's going to be an addition of 3 bases added, so it's 2 C's and A's at that 3' end, and then there's also some splicing. So some parts, there's also base modification where the bases can be modified, and then splicing where some of the bases are removed from the transcript, forming the mature tRNA. There's also modification of rRNA. So rRNA is made into sort of a long molecule, and it undergoes methylation at these specific sites. So those specific sites represent what's called the 18S, 5.8S, and 28S. And after it's become methylated, it'll then become cleaved to form these 3 different rRNA fragments. And this trimming is actually directed by these ribonucleoprotein complexes that contain another type of RNA, regulatory RNA, called small nucleolar RNA. So it's located in the nucleolus, and these small nuclear RNAs, also known as SNO RNAs, are involved in the trimming of the mature tRNAs. Okay, so we're going to focus most of our discussion today is on the modification of mRNAs. This only occurs in eukaryotes. It includes adding this, what's called the 5' cap, adenylating the 3' ends, and then splicing of coding sequences. Quick drink of water. So I'm not going to test you guys on the modification of rRNAs or tRNAs, but just giving you the information. But I will on modification of mRNAs. Okay, so one of the things that has to happen to an mRNA is this addition of what's called the 5' cap. So this here represents essentially like the end of, or I shouldn't say the end, but the beginning of like the mRNA molecule, and all of this is, this represents here the beginning of the 5' end of the mRNA molecule, and then this represents the 5' cap. So a lot of times there's what's called this this 5' cap that's added. It's a 7-methyl guanosine that's added, and it's attached by this 5' to 5' triphosphate linkage. The hydroxyl group of the riboses of neighboring residues are methylated, and together this 5' cap helps to prevent the mRNA from degradation by exonucleases. Another modification that happens is essentially the maturation of the 3' end. So it's going to involve trimming the 3' end as well as polyadenylation. So at the 3' end, there is what's called a polyadenylate tail that is added to the mRNA transcript. And this poly-A tail is important because it helps to also protect it from degradation from nucleases and phosphatases. And the way this works is that you have this large complex of protein that's actually recruited, so this represents a large enzyme complex that's recruited to Pol2 when the polymerase encounters a specific RNA transcripts, which terminates transcription, and then Pol2 will fall off, and then this enzyme complex is still bound to the mRNA transcript. It will then cut the 3' end with its endonuclease activity, and then add this tail of A's that's catalyzed by polyadenylate polymerase to then add the poly-A tail. And so because all mRNAs have a poly-A tail, often that poly-A tail can actually be used as a way of actually isolating mRNAs. For example, you can purify mRNA by running it on a column that is attached to a bunch of T's. So that's what like a mechanism for isolating mRNAs. Okay, and then the last thing we're going to talk about is the splicing of coding sequences. So after transcription, we have our introns. Remember, that's sort of the non-coding part of the RNA molecule that must be spliced out. So that way it only encodes exons. So splicing is going to remove the introns so that way you have exons. But the other thing that's important for splicing is you can get alternative transcripts. So you can have alternative splicing that only has that results in transcripts that only has certain exons in the mature RNA message. And so the mechanism so we're going to go into the mechanism coding sequences. It's in terms of the splicing, it's performed by nucleases and ligases, as well as ribozymes. So if something is a ribozyme, it's an RNA that has catalytic activity. And so some RNAs that become spliced, they are self-splicing. They are self-catalytic. Whereas some RNAs, they're not self-splicing, and they require what's called the spliceosome. So this is sort of a huge ribonuclear complex that's composed of proteins and then small nuclear RNAs that sort of help to catalyze the splicing reaction. So we're going to go through some of these types of splicing mechanisms. So one type of mechanism or one type of RNAs are called these group 1 self-splicing RNAs, meaning they do not require these spliceosome components, and the RNAs themselves are catalytic. And so the way that the splicing occurs is you have here's your primary RNA transcript. So here this is the 5' exon, here's your intron, and then here's your 3' exon. And remember, the goal here is to remove the intron sequence. So the way this works for the group 1 self-splicing mechanism is you have a free guanosine, and the 3' hydroxyl group that's on the free guanosine is going to attack a phosphate of a base that's located in the intron. And so this is what it looks like specifically. Here's your 3' hydroxyl group. It's going to attack that 5' phosphate of the intron, which will then displace the 3' hydroxyl group of the 5' intron. So now we have that free guanosine attached to the intron by a 3,5 phosphodiester bond. And then in the next reaction, the hydroxyl group, so the 3' hydroxyl group of that 5' exon, then attacks the phosphate of the 3' end of the intron, then displacing that 3' end of the intron. So then you have a 3,5 phosphodiester bond between your 5' intron and your your 5' exon and your 3' intron. So that's the mechanism by which an intron is removed using the group 1 self-splicing mechanism. Okay, so that's the group 1 self-splicing mechanism. Again, it's going to use this free guanosine. For the group 2 self-splicing mechanism, rather than using that free guanosine as the source of the nucleophile, the source of the nucleophile in the group 2 self-splicing mechanism actually is the 2' hydroxyl group of an adenine that's actually located within the sequence of the intron. And so it's very, very similar to the mechanism of the group 1 self-splicing RNA, except, again, it's going to use that 2' hydroxyl group of an adenine that's located within the intron. So the 2' hydroxyl group is going to attack the phosphate of the base at the 5' end of the intron, which will then displace the 3' end of the 5' exon. This creates what's called a lariat structure, where we have a 2,5 phosphodiester bond between the 5' end of the intron and that 2' hydroxyl group of that adenine. And then in the next reaction, the 3' end of the exon will then attack the 5' end of the intron, displacing the 2' exon, displacing the 3' end of the intron, so that way now we have a 3 to 5 phosphoester bond between the 5' exon and the 3' intron. Okay, so those are the group 1 and group 2 mechanisms. The group 3 RNAs, these are not catalytic. So they do not have self-splicing activity, and instead they require these spliceosome ribonucleoprotein complexes to help catalyze the reaction. And so it involves these small nuclear ribonucleoproteins called these U1, U2, U3, etc. They come, they bind to these sequences of the RNA. So the RNA themselves, it can be, the way that they're actually detected as the group 3 RNAs is that they have a GU at the 5' end of the intron and an AG at the 3' end of the intron. And so these spliceosome components will bind to the RNA and help to catalyze the reaction. The one thing about this type of group 3 RNA is they actually use a very, very similar mechanism of splicing as the group 2 RNAs, in that it actually uses an adenine that's located within the intron. So it uses that 2' hydroxyl group of the adenine to act as a nucleophile to then attack the 5' end of the intron. And again, it's going to form that lariat, which then is going to be spliced out by attack of the 3' end of the exon to the 5' end of the 3' exon. Okay. All right. Okay, and then just one last thing is the RNA-dependent synthesis of DNA and RNA. So we talked about transcription in in the context of RNA polymerase. There are other mechanisms by which transcription can be brought about, and so there are viral enzymes called reverse transcriptases that can synthesize DNA based on an RNA template. And so actually this really isn't, this is transcribing RNA, transcribing DNA by using an RNA. And so some of these viruses include HIV that uses reverse transcriptase. And so when the mRNA is transcribed into a DNA, it produces what's called a cDNA, and typically this is shorter than that of an mRNA. So, and usually it does not contain any introns. Okay, so with just the last few minutes we have left, I'm just going to go and show you guys this movie, and then we will get on to the group presentations. And I didn't bring a speaker, so sorry about that. I just did not have arm space left. It's a pretty cool movie.
Workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1.
You guys notice the DNA touching the
blue molecule
polymerase?
racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When
All right.
the RNA copy is
I'm
complete,
going to stop
it
that
snakes
because we need
away
to
from the nucleus and into the outer part.
move on to our group presentation. But I think it's a pretty cool group. Okay, so we have our presenters come up and do their presentation. You guys want to go first? All right. Do my. Okay. Probably just need a laser and that's it, because I know you guys have your